My interest in cognition and behavioural neuroscience actually started very early in sort of high school. I was really interested in psychology. So as an undergraduate, actually, I did psychology and I majored in both psychology and neuroscience. And then that forged into a PhD, which looked at changes in brain connections and how they underlie learning and experience. And that led me to start a postdoc and obviously have the great chance to be in the UK to work alongside Lisa Succeeder and Tim Bussey at an incredible time when they were really developing the touchscreen-based methods. So it forged my passion in understanding the brain changes in connections that underlie cognition and learning, and so hence my work with Seth Grant and how we forge that into advanced preclinical models that enable us to really be able to test that. And so during that time, I must say it was phenomenal. Some key components of that were not only being involved in developing those initial prototypes, but the team-led Nature Protocols papers that subsequently came out to really instruct and shape the various tests that are available to be able to use within the touchscreen setup. And then, of course, there was the sort of real instrumental work that we led, really showing the clinical translational utility of these approaches, how we can utilise these tests in rodents and use analogous tests in humans. And in some cases, you know, the dream of using the same test in both species. What can I say? It's in the time since their development and commercialization, I think there's been enormous strides in really refining what is available. Obviously, when we first started out, I recall thinking very deeply about looking at the various components and how we refine those, including things like the sensitivity of the actual touchscreens, how important that is to ensure that they accurately measured the responses. Additional parts since that initial prototype is how the changing of the walls from, a square to a trapezoid or cone, which, really again helped focus attention for the animals and how we best capture what it is that we're trying to do in these assays. And then obviously now with the newest generation, I think what is incredible is advancements in how you can integrate multiple chambers in a more seamless design. So I think what has been really lovely to see across the years is there's been a continual efforts placed on the real strong foundations of the system, but continuously refining how we modify those to ensure that these are, again, staying cutting edge in terms of ensuring that we are capturing the most appropriate and best measures to get complex behaviour. I think, what really drew me to it, and I was so inspired, is really the key goal of how is it that we develop tests that capture the same cognitive constructs in humans. I think this is the key. It's really about how we boost translation. And I think the touchscreen assays have very much addressed this fundamental need. And so I think back to some of the really core tasks that were developed across years in the Bussey Succeeder lab. We were dealing with a handful of those assays, which again, as I alluded to, we published in the Nature Protocols. But since then, what can I say? I think there's been a really large community, labs such as mine, including Lisa and Tim's lab, who continue to, and many others that have really come together to develop so many more additional tests, again, with the goal of really trying to capture and measure the diverse cognitive constructs that are relevant for humans and that are fundamentally disrupted in brain disorders. And so, it's been really amazing to see the growth of that. We've got above 14 tasks, as I know, and these continue to grow. And I think that is because of the investment from the community that is, very much paying it forward as to how we continue to refine and develop these tools. Yeah, I think, one of the great things about the approach is since it's been commercialized, I think it is a truly global tool. So many laboratories have access to this. It is very much plug in and play in the sense that the components are all ready to use for people, and I think that makes a huge difference. The nature of how they're actually set up enables labs to be able to integrate those and hit the ground running. I think fundamental to this, though, has been how we bring that community, how we give access to many people, but also then train people into how best to analyze and interpret the data that you get. Because I think one of the incredible things about the system is its rich data that you can get from the various tests. And so I think that the learning part, and this is where the community continues to refine its interpretation, is how you ensure you take that rich data and create meaningful outcomes in those. I think cognition is so complex. In animals, it really relies obviously on aspects of movement. So I think what's been great is, people thinking deeply about how you can start to parcelate that out. Are you really tapping into the cognitive and behavioural outcomes that you're really wanting to investigate, and how do you separate that from other components that may not be, what you're looking for? So I think it's a valuable tool that has really very much increased access. And access in a way that people can generate data that is obviously far more comparable across, sites and laboratories. There continues to be, increased focus on reproducibility in neuroscience. This is something we have to be mindful of. And I think these assays have really helped towards that. There are standardized protocols, but along with that comes, I think there is this education of the community in terms of how we do behavioural neuroscience in a more refined and robust way. So I think it's been a great advance in how we drive preclinical discovery science to have greater translational impact. Yeah, things have just moved. So as I shared when I started working with these the touchscreen system, was really deeply and have always been deeply interested in behaviour and cognitive behaviour and what's really happening in the brain. We continue to work with a variety of tasks, which again map onto clinical domains. I have a personal interest in looking at psychiatric conditions, particularly complex psychiatric conditions like depression and schizophrenia. And one of the advances I think that has happened, and it's very much available now, is the integration of the behavioural tool with in vivo recording approaches. So whether that be fibre photometry, optogenetics, cellular calcium imaging, to look at single cell resolution. Some of our work has really capitalised and leveraged these more modern neuroscience techniques. But these modern neuroscience techniques are only really valuable if you can pair those with a real foundation in the behaviour, which I think the touchscreen chambers enable. So we've been really excited because we've looked deeply into better understanding, for example, the reward system in particular, understanding how cortical control, predominantly, for example, the prefrontal cortex, how the prefrontal cortex function can really shape some of these cognitive domains that are really relevant and disrupted in psychiatric conditions. So I think that's the new path forward. We're really excited. We've got some great data that is currently being generated and will be available. And what I really hope is it's the next iteration of how we take these really, phenomenal, fundamental preclinical assays and then merge them and stay very cutting edge in terms of the more broader neuroscience tools that are available that are giving us, real understandings into what are the neural changes that underlie cognition and complex behaviour. I think, mental health is something that is so dear to my heart. And a challenge for the field is really around how do we develop better treatments for people. We know that there is no one size fits all. We know that there are treatments that are currently available, but these don't work for everyone. So we need a wider toolkit to make it really appropriate for, and so that everyone has access. And part of that is how do we, better translate, how do we develop new therapies? And this is where I think the preclinical research is absolutely fundamental. So the translation of preclinical to clinical has been something that has been my deep passion. And this is where I think the touchscreen approach has been absolutely fundamental. So I think we have made enormous headways, and we continue to, in terms of really refining the preclinical approaches that we have and available to be able to, really tap into those symptom domains that are impacted in mental health, and thereby not only understand what's, the neural basis or the biological basis of those, but have a way in which to more effectively examine and develop treatments that are actually going to be effective. And I think this is very much happening on the ground right now. And I think over the next 10 years, we're going to see major strides in how that preclinical approach really advances that clinical translation.